Welcome everyone. Today we're going to be talking about advanced programmatic access to UniProt using Python. Obviously our examples can be translated into other programmatic languages as well. So if you have an alternative, please feel welcome to take the code that we're providing and translate that too. So as Flaminia said, I'm a curator at UniProt and Aurelian is our website team lead. So how we're going to work today is I'm going to show you how to do something on the website, and then Aurelia is going to run with the Python and show you some examples of how you can access that data programmatically. So if you don't know, UniProt is a biomedical research support resource. We provide a rich resource of freely available, stable, comprehensive, and always accurately annotated protein sequence and function information. We primarily focus on primary sequences, which include isoforms, as well as disease variants. And we always focus on physiological function of a protein and annotate information such as subcellular location, pathways, reactions, interactions, as well as involvement in disease. We have a wealth of structural information, including topology and access to 3D structures, and we also have a set of protein analysis tools such as BLAST and sequence alignment. UniProt itself encompasses a number of resources. So here you can see on our homepage, we have a set of 4 main resources. We have the UniProt knowledge base, known as UniProt KB, proteomes, UniRef, and UniPark. And today we're going to touch on UniProt KB and also introduce UniPark. So today's aims and outcomes are to identify different ways of accessing UniProt data programmatically and how to pick the most appropriate access methods for your workflow, to filter our entries by annotation type or your target characteristics of interest, to explain links between different UniProt services, in this case UniProt KB and UniPark, and also where to find documentation and a helpful user guide for your programmatic access of our database.
So we're going to jump into how we can access UniProt data. So the focus of today will be the API, but we have also other ways to access the data depending on your specific use cases or the scale of data you want to access. So most of the case, if you want to download, if you want to do big one-off downloads of nearly all of the data in UniProt KB or in UniProt, and if you're also able to do some post-processing, then the way to go would be to access the data through the FTP. Those are pre-computed files that you might be able to access. You have everything in FASTA format or XML format. So you have access to all that data there. So for something that would be more medium size, so not the full data set, but specific queries and all of that, or if you want to customize the data before downloading it, then the API would be the way to go, and we will see how to do that today. And if you have smaller size downloads or things that you can do manually, a few pages that you can do that manually because you need that just one time, then maybe browsing the website and downloading directly from the website would be the way to go. We also have the case where you might have to do a one-off download, but of still a big amount of data. So there you could also see that on the website, if you are trying to download more than 10 million items, so 10 million entries, you will be offered the option to do that asynchronously. So the website will generate that file in the background, and then you will be able to come back to it later and download it. That's again through the website. So the focus of today, as I said, is the API. So to access the API, so you should have a look at the documentation all the time if you want to access the API, because really that's listing all the different capabilities that the APIs have. We have different endpoints in there. So we have, for example, UniProt KB and UniPark, which will be touched on today. We also have some other endpoints related to the other data sets within UniProt. That could include UniRef, clusters of proteins, and proteomes, as well as other data that you can see on that screen. We also have tools, which is ID mapping, part of the main UniProt website API. We also have BLAST, Align, and peptide search, which are available but not provided necessarily by the ABI UniProt team. We'll see an example of a line later today, but it's not provided by our team. It's provided by another team at DBI. Just a few basics on accessing data. So you will be sending requests to a server, and the server will respond with a status code. So anything that is in the 200 range means that it's all fine, it's all good. You have your data in there. If you see a 400, it might be an error on your side, so potentially a wrong URL or wrong query. So this is something that you need to check and debug. and you will want to have a look at the error message and also the help page. And if you cannot really sort it out, then you can also reach out to our help desk. If you see a 500, this is an error on our side. The service is having issues. And so most of the time, this is not something that you did wrong, but this is something that is temporary. So we would recommend to just wait a little bit and retry a few hours later to see if if the error is still happening. We should see it, but if you see that the error is persisting, please feel free to get in touch with us. In the response, you will get data, which is the important thing that you want to download, but you also get some metadata. And some of this metadata, it will be returned through the response headers. And so we will see that in the examples in the demo. But the important one that you might have to have a look at are the total number of results. So this is not total number of records in your query, but you also have the UniProt release, which can be quite important to keep track of when you download a specific data set, and a link to paginate. We'll see an example of that, as well as content type to see the type of content, if you have like XML, JSON, FASTA, or anything like that. So we'll jump to a live demo. We will be sharing the link at the end of the demo, so don't worry about that. Just focus on following the demo, and you will have access to all the code just after it. So there's no problem there. This is using Google Colab notebooks. The same code can also be run through Jupyter notebooks if you're more used to that, or directly in Python code or in the IPython CLI, as well as within Python scripts if you're writing scripts yourself. And as Amy said before, this can also be applied to other languages. Obviously, you need to adjust the code, but everything that we show is not really limited to Python. It can be used in any language that is able to do requests on the internet.
So,
okay, so sorry.
that's all right.
So I'm just going to show the basic setup. So we have here some code with some variables. So we will be using the website API, which is hosted at rest.uniprot.org, and we'll be using specifically the UniProt KB and UniPark endpoint. The documentations are here. So in any case, you should always have a look at the documentation before trying to use the API. And also here I have a helper function, which is just in charge of doing the request. And if there's any of those 400 or 500 errors that I mentioned before, we would be able to see that information on the screen. Hopefully we don't have any error today during the demo, but you never know. And so the basic search, so if we search for everything, so star search here, this is searching for everything on UniProt KB, then you will get some data sent back by the server. And so what I will have a look at here is the number of results within that payload returned by a server, so the length of the results array. And I will print this, as well as I will print all the different headers that are written by a server, just to show you in an example what we've seen in the slide just before. So here you see number of results 25. This is actually normal. Even if you download, even if you request everything in UniProt, we will never return to you the whole thing at once. You will be returned a page of results. So here, this is the 25 first results. It will be your responsibility, your code's responsibility to then iterate through the different pages to be able to access all the data that you need. So, and the headers that you see here, so the ones I wanted to highlight is the API deployment date. So when the API has been deployed, 9th of February, the UniProt release that it corresponds to. So this is the data for 2026-01 release. And so after the next release, you will see that this will have changed. The X total results, so this is the total number of results for your query. So this is actually the numbers that you will see on the website of 203 million entries in UniProt KV. And again, the release date of that data set, so 28th of January. Let's go back to the slides, the other way around. There you go.
There we go. Okay, so I'm going to show you how to do our example that we're going to follow through with on the website, and then we're going to look at that programmatically, and that's how we're going to do the rest of the webinar. So as part of our website, when you search UniProt KB, we have an advanced search functionality, and that allows you to do controlled searches with different search fields. So this is an example of doing a free text search for parking. So this searches all the entry, whether it's the protein name, gene name, function, any section of the entry, we're searching for that piece of free text. So in this case, parking. And I've also brought in there a taxonomy filter for human. So when you click search on the advanced search, you're going to have parking and taxonomy 9606. So that's going to give you a results table with 151 results like here. You can customize that results table. So yours might look very different to mine, but that's the one I'm running at the moment. So you can see we've got the accession ID, then the entry name, protein names, organism, and gene names in mine. We're going to open a protein entry. So I'm going to open human parking, which is the top entry there. So you do that by clicking the accession ID, and that opens the protein entry that you can see here on the right. So the very first information that you see in a protein entry is its identity summary. So you have its accession ID and its UniProt kind of identifier. You have the protein name, the gene name, whether it's a reviewed or unreviewed UniProt KB entry. The organism, we search in human, so it's human. The amino acid length of its canonical sequence, and then its protein evidence and its annotation score. Under that, we have a selection of tabs with more information in, and then typically at the top of a protein entry, if we've got function information, that's what you're going to see first.
So now how to do that using code, so using the API. So everything that you can do on the website, there should be a way to do that programmatically, and we'll see that now. So that search that we had before, parking and taxonomy ID 9606, the same one that we did on the website, we can do the same one on the API. Then what we will do is have a look loading that data, because I know it's in JSON, so I will pass that as JSON. And then we'll have a look at those headers that we mentioned before, the total number of results, the number of results in my payload, as well as some other information here. So if I do, I'll comment that first, and I'll run this. So I see that according to the headers, I have 151 results, same thing as the website. And the page total, so the number of results in that page, in that payload, is 25, which is expected, as I said before. And if you have a look at the link, especially the linker will tell you where is the next page. So here, next page of result, you will see that you need to access the data through this URL. So you don't need to do that manually. You can do that programmatically. And so for that, you will need to do that in a loop. So for as long as there is the next page, so as long as the result, the server is telling you that there is another page of results after this one, you will be able to get that new URL and then access that data programmatically. And so we'll do that in a loop, and it will stop as soon as there's no further page. So let's run this, and you will see that we'll get data one after the other. So first page over the 25, 25, etc. And the last page, obviously, we have 151 results in total. So the last page will only have one result in there. Okay, so this is using pagination. So we have 151 results. You also can do that through the Stream API. So here the advantage that you only do one request with all of it. It will get the whole result set. You do have trade-offs though by using this approach, is that it's harder to follow progress. So here for 151, it's not too big of a problem, but if you want to have thousands, potentially hundreds of thousands of entries, then you will get the request, you will get results back, but you will not be able to know where you are in your request. Also, if there's any failure during the download, then you will need to restart from scratch because you cannot resume the Stream API. You will need to redo the query from scratch. Whereas if you had an issue doing pagination, then you can see that if page 50 fails, then you can retry page 50 and then continue. And also, it's not sorted by score, so you will get all the results, but not in a specific order, and also not in a deterministic order. So if that's important to you, the Stream API is not will not be helpful. But if it's not important to you, this is worth considering. And you can also explore the data in different formats. So by default, we have JSON, but here you can see in all the examples, those are commented out. So afterwards, feel free to uncomment and play with them. But you can download that in XML. In this format or in FASTA. I can show you quickly the FASTA one. If I run that, then I will get all the sequences in FASTA. And then the last one, which is in TSV format. So TSV for tab separated values. And so this is more of a tabular format, which would correspond more or less to the website view that Emily showed before. You have entry names, status, etc. And just as in the website, you will be able to customize the output. So let's have a look at that. And say, here, I want the column, so I will require specific fields. I want ID, accession, length, and a comment about subcellular location. And if I run this, then I will get this table of results for that query, parking human, in that specific order. And then I can add or remove some or reorder them, and the result would be adjusted according to what you specified here. Finally, to retrieve one single entry, so just like Emily clicked on the top result and opened the page for that protein, you are able to access data about a specific protein based on its accession. So if I have the accession, I'm not using the search endpoint anymore. I'm directly passing the accession and retrieving the results. So if I run this, then I will get by default in JSON, I will get an object with a lot of information in there. Everything that you see on the website will be there, but you can also customize it. So if you're only interested in, let's say, subcellular location information, we'll be able to customize it this way. And if you run that, then what you get is a smaller object with some basic information about the entry, but then all the rest of the information is only about subcellular location information. Okay. And back to the slides.
So you might notice in that bit of code, we've got some echo codes that explain where that information comes from. I'm going to show you how that looks on the website, and then we're going to look at it programmatically again. So as you navigate the website and explore the website, you're going to see these little evidence tags, which are here in gray. They're highlighted with these red arrows. We've got a range of them. We use it to describe where the evidence for a piece of data comes from. So you see here in names and taxonomy, we've got a tag with 2 publications. And if you click on that, it gives you kind of like a scrolly bar with basic information about both of those publications and links to PubMed and Europe PMC. And lower down for the alternative name, we've got an imported evidence tag that shows you that the evidence for that particular alternative name for parking comes from HGNC. So that's a cross-reference database. And then on the right, we've got a subcellular location section of our website, and we've got numerous tags here. The evidence tags that point to publications, again, are all expandable. A publications tab basically means that's experimental evidence. A curator like myself has read that and attributed that tag. And then further down for mitochondrial outer membrane, we've got a similarity tag. So when you open that tag, it points to an ortholog. Here you might find in that ortholog experimental evidence saying that that protein localizes to mitochondrial outer membrane, but we haven't got experimental data for the entry you're currently in. So you might be in human, but the experimental evidence might be in mouse. So we've propagated that information over because orthologs are conserved. So that's how we show those tags on the website. This is what they look like when they're not obviously graphically shown on the website. They're a control vocabulary evidence tag or echo code. It's basically an ontology. The echo codes that we commonly use are listed here. So 269 is experimental evidence, again, manual curation of a paper. The data says this protein does this job or is localized in this tissue or in this part of the cell. And you will see the PubMed identifier linked in there for where that evidence is from. Then we've got 250, that's the sequence similarity tag, shown as by similarity. That's when you've got evidence in an ortholog. So you might have the experimental evidence in the mouse entry, but no experimental evidence, let's say, in the rat. So you'll see when you click that tag, the UniProt accession of the mouse entry, the entry where the experimental evidence is. Then we've got 305, this is curator inferred data. So this is usually followed by a PubMed ID as well in most circumstances. And this is when a paper has experimental evidence for something, but it might not fall particularly in our very stringent guidelines for curation. But the curator in this case has a broader sense of knowledge of the area of this particular bit of research or this particular protein signaling pathway or protein family, and they can say that based on the evidence in this paper, we can decently confer that that is a conclusion that we can be made from that paper. And then we've got 312, and this is imported evidence from an external evidence source. So we have a range of over 800 cross-reference databases. There's a list on our website, and essentially this tag is used to say we've imported that data from an external database. You click on this tag and it will tell you the link to the database we've imported that from. There's a full list of codes and their explanations at that URL there. And there's some examples of the tags on the right. So When you look at this on the website, you're going to see this is a subcell location of parking. You see we've got the graphic on the left that shows you whereabouts in the protein in the cell that the protein is localized to. And as you hover over each one of these terms, it will highlight that section in the cell as well. And you see we've got a real mix of different experimental and bisimilarity tags here. So the cytoplasm at the top has 11 publications that provide evidence that it's localized to the cytoplasm. And then the mitochondrial outer membrane is actually a bisimilarity tag to the mouse in this case. So you can see within each comment of a protein entry, you can have a mix of evidence tags. So we're going to show you how you can see them programmatically and also how you can filter by them too.
Okay, so let's jump back to the code. So we have those evidence codes. So again, we start with that entry and we focus on subcellular location data. So if I first have a look at what I have in there. So if I look at my comments and focus on the subcellular location information, after doing that query, that you will see some evidence codes in there. And so you have multiple subcellular locations, and for each subcellular location, you might have one or multiple evidences with different evidence codes. So we will have a look at those and print them a bit nicer. So if we now loop over all the different subcellular location information that we have in the payload, we will just decide for now to play the ID. So that would be the UniProt KB, the UniProt internal ID for that subcellular location, the value, that was something that is more human readable, as well as the number of evidences. So if I run this bit of code, you will see 8 subcellular locations with those names, as well as the different numbers here that correspond to the number of evidences, which is exactly the same thing as you can see on the website. You have 11 publications for that cytoplasm, and etc. So now what I can do, since I have tested that programmatically, is do a bit more advanced logic. If I wanted to, for example, focus on the 269 evidence code for created publications, then you will be able to, for example, it's just an example here, you should probably adjust that code to your specific use case. But if any of the evidences within the list of evidence for a specific subcellular location is equal to 269, then I will display them. So now, instead of having 8 results here, if I run this code, I will see only 5 results, which correspond to which will have at least one evidence that contains 269 evidence code. Back to the slides.
Okay, so now we're going to talk about how to retrieve and compare orthologs on the website. So I'm doing a really broad free text search here, not even an advanced search. I'm just searching for parking. That's going to pull back everything where parking is in any section of the entry. So it gives me nearly 274,000 entries in our results table. I'm really only interested in this case in a few model organisms for parking, also known as E3 ubiquitin protein ligase parking. A lot of you will know it's involved in Parkinson's disease. So if you want to retrieve and compare the sequences of these orthologs, you select the ones you want using the ticks on the far left of the results table. I selected Drosophila and human, rat, and mouse. And when you tick them, you can then add them to basket. And your shopping basket, it can act as just something to save protein entries for later, which is exactly what I used to do when I was busy running around the lab. Or you can use it to actually run analysis tools like we're going to do today. So in your basket, and you can find the link on the top right of the homepage, in your basket, you're going to have your pre-collected proteins. You can select tools and then align.
Sorry.
Oh, we're skipping ahead. You can select tools and then align. What that will do, it will open an align form that will be pre-filled with your sequences. In this case, these 4. You click run, and then that job will appear in your tool results bar, which opens automatically. When it's completed, you will see it will show complete and a little green dot. And when you click the completed, you get your results like you've got bottom right here. What this is essentially showing you is a similarity between orthologs. The darker the purple, the more conserved the residue. So you see you've got blocks here of really conserved residue stretches. And you can see the Drosophila entry here has got a completely different end terminus to the three mammals sequences that we selected. What you can also do with your align results on the website is you can kind of build into this the sequence annotation that we've got in the entries to see if those residues are conserved between your sequences that you've queried. So to do that, you click on the previous slide. I'm going to show you this. There's a little red circle there, and it says select annotation. Bear in mind that when you're selecting annotation, you're selecting an annotation specific to a sequence, not to all four of them in this case. So I'm going to select POC in human, and I'm interested in this case in the active site. So you use a drop-down box, you select your sequence of interest and then your particular annotation of interest. And what it will do is it will highlight that residue or kind of chunk of residues in red that you find of interest. So in this case, it's a cysteine. It's got a red box around it, and you can see it's conserved across all four of our sequences because the active site is conserved across the model organisms. And what you can also do is you can click that cysteine, and it will tell you exactly where the evidence for that annotation has come from. So here I've clicked the human cysteine, so you can see you've got a sequence annotation from a pro site rule saying this is an active site, and then you've also got evidence from a publication. And if you were to click the cysteine for Drosophila just above that, those evidences are different, and same for rat. So you can see where the differing evidence has come from for that particular annotation across those entries.
So how to do that programmatically? Okay, there we go. So this is where we will be using a different API. So here we will get the data, the sequence information, and the data about the active site from the UniProt KV website API. But we will send that to the Align service, which is one of the tools that is provided by the EBI for all to be able to use. And so we will be using that different API there. So I'll show you that in a moment. So the four accessions that Emily had selected, I have them in a list here. And so I will create a query that will give me just these four entries in a table. So I will query for accession this one, etc., etc. Actually, it's missing the O at the beginning. Just a comment, so that's not going to affect anything in the code. But so I'm going to generate that query. I will query the endpoint with that, and I will request the FASTA format. Then, so let's have a look at that first. I'm not run the code just yet. So if I run this, I will see the FASTA format, which is the input that is needed for the alignment. I need the sequences to be able to align them, obviously. And then if I have a look at the data here, so I have the data in JSON, so same query, but I will ask only for the active site feature field. And if I have a look at that, you will see this information here. So a list for all four entries, the information about active sites. So you can see that on that specific one, it's at position 431, but for another one, it might be at a different position. So here, for example, 449, which is why it's important to align the sequences just to make sure that we are actually looking at the same ones or not. So here what I'm doing is sending that request, that specific request with the FASTA file to this endpoint. This is documented here. So if you want to have a look at the API documentation, follow that. And I will need to pass this information. I want the output in cluster num format, and I want the order to be the order of the alignment. Then this will return to me a job ID. So if I have a look at the job ID, it's just an internal job ID for the results. And then with the job ID, I will be able to ask for the status of that job to the server. Because this is an asynchronous job, basically you will need to ask the status until it is finished. So if I run all of this at once, I will get first a job ID, and that you can see because I asked for a status immediately, you can see it's queued. But actually, if I run that code again now, let's run this bit of code now. You can see it's finished because alignment are usually fairly quick to run. And so now that I know it's finished, I can access its result. So now I have this endpoint, which is based on a job ID, I want the result. For for that alignment. And then if I print this, you will see the alignment result. So here you have a similar view as what you have on the website, because the website is using the same output here. And instead of having colors and all of that, you will have the similarity based on those symbols here. So this is just a cluster specific. There's nothing specific to UniProt in there. And based on that, then you can try to map the active site information to the alignment. Go back to the slides.
Okay, so now I'm going to briefly introduce UniPark. UniPark is another resource that UniProt as a consortium and as a resource, as a database provides. UniPark is a sequence archive. So think of it as like a sequence library instead of a protein library. So instead of having an identifier for every protein entry, now you've got a UniPark identifier for every sequence. Every sequence is unique, every identifier is stable. So within a UniPark entry, you will have the sequence. You can still use tools, blast and align for that sequence, and look at its feature view and structure view as well. But this time, instead of annotations, you've got cross-references, and the cross-references detail other identifiers and entries that are mapped to that particular sequence. So these will be 100% identity mapped. So here you can see we've got some UniProt KB entries, and if you were to scroll down there, you'd also have some ensemble sequences linked and some ref sequences linked as well. It will contain multiple database sources as well as sequences from various species if they're exactly the same as one another. And you can find proteins here that have been removed from UniProt KB or sequences that haven't been imported to UniProt KB either. So what's the use case for UniPark? There might be quite a few of you that have never used it before. So we've got big changes coming to UniProt in the next release, and quite a few of you might find that some of your proteins will be in UniPark rather than UniProt. So here's an example of when you don't get any results in UniProt KB. So we're doing an advanced search. We're looking for the protein name chemotaxis protein key A in a very specific strain of E. coli. So it's a substrain of K12. It's telling you, sorry, we've not found any results in UniProt KB. But do you want to do the same search in UniPark? And that link's in blue. So if you click that link, you're going to get the exact same search. But this time you see we're in green. We're doing this in UniPark's resource, and you've got 4 results. So what this has done is it's searched for the protein name within the UniPark identifiers as well as a particular taxonomy. So we've got 4 results. You can see we've got different sequence lengths. And in this case, we're going to explore the third one down because it's a really nice example of how, when you've done a search in UniProt KB and instead of getting a protein entry back, you get nothing, no annotations. We've taken you to UniPark, and within that UniPark identifier, you see we've got a couple of reviewed UniProt KB entries. And in this case, these are for K12 strain of E. coli, the reference proteome. So you can click these entries, you can go to their pages, and you can see all the annotations on there as well. And you've still got all the tools where you can do your sequence alignment and blast search for these as well.
So let's have a look at how to access that programmatically. So let's do the same path, the same steps that Emily followed there. So we have a query, which is that protein name and that specific taxonomy that correspond to that substrain of K12. And first of all, if I do a search in UniProt KB and I have a look at the data, then if I run this code, you will see that, as expected, you have no result. The same as the website. You have no result at all. So here you don't get the nice suggestion just like the website. You will need to know that you can try again same query in UniPark and see what you get there. So if you get that, if you do that same query in UniPark, what you will get is a list of results. Here you don't have UniProt KB results anymore. You have UniPark results. So the format will be slightly different, but I just want to show you what type of information we have in there. So I'm just going to scroll to the top. You can see there's a lot of data. Let me try to get the scroll bar out of the way. Okay, there you go. And so the first result I have is the UniPark ID, the same one as you could see on the search result on the website. You can see that the common taxon information is there, the different UniProt KB accessions that have the same points as that UniPark, the sequence itself, because you might be interested in the sequence, and as well as some sequence features. We have for each of them sequence features from InterPro that have been annotated there. And if they're based on a sequence, then you will be able to access them. So you can see this domain information, etc., from the different member databases of InterPro. So what can I do with that? Now that I have this, I can also, oh, sorry, I just wanted to show you also that I can get the FASTA. If you modify the format, you have FASTA, so you have the 4 UniPark entries in there. So if you wanted, for example, to align them using the same flow as we had before, then you get the FASTA, you align them, and you could also potentially add some other sequences that you might have just to be able to see what is conserved or what is not. And if you want to get the information from the UniProt KB accessions that have the same sequence as Emily has shown on the website, you have that information in there in UniPark, and you also have that information in the payload there. You can then access the UniProt KB information from the UniProt KB endpoint, and then be able to analyze if the annotations that you have there, that you found there, are meaningful to you or not, or if they can be applied to your sequence with your specific strain of interest. Then go back to the slides.
So I'm going to show you another example of how you might end up in UniPark when you're expected to be in UniProt KB. So let's say a few years ago you did a mass spec experiment, you mapped to UniProt accessions, and you find this really interesting protein, and you're reviewing your data and you can't remember what this protein did. So you put the accession back into our search for UniProt KB and you get a results table and it says we deleted this accession. That's fine. You can still click that accession ID and it's going to take you to a UniPark page. So you've got the UniPark identifier that links you to that specific sequence. You've got the accession ID that's linked to that source, the protein name, gene name, and it's also got the annotations that will be automatically annotated to this entry as well. And if you click the UniPark ID, you'll also see other entries that we've got in UniPark that are similar and associated with this identifier as well. This view might change as we go forward. We're still adapting UniPark for its future uses, but the facts remain the same. You're still going to have the tools. You're still going to have the annotations that are available. You're still going to be able to review your old UniProt KB accessions that are found in UniPark, and you're still going to be able to link that to a UniPark identifier as well.
Okay, so now we're gonna see how to do that from the API. So this is the use case of I have an accession. I want to get information about that accession. So the first thing I'm doing is a request to the to UniProt KB, just to see that the endpoint will tell me that this is an inactive entry. So this is not the end of the road. You can actually explore the information. So first of all, you have some information there. It's been deleted. Okay. Why? Because it's a redundant proteome. So you can have a look at the help page to see what that means. You might have different deleted reasons. And you can see here the UniPark ID. So with that UniPark ID, you'll be able to search in UniPark for that UniPark ID and get all the information that you have there. Also potentially other UniProt KB that have the same sequence. And so all of that is through the UniPark API. So just from the output of the UniProt KB data, I will be then extracting the UniPark ID, and I will pass that to the UniPark API. So UniPark API / the ID that I'm interested in. And then if I print that, you will see that I get one result in there, which is the result the payload for that UniPark ID. Let me just again go back to the top of data. So I have to scroll a little bit. Yes. Okay, we're back at the top. And so this is the UniPark ID that I saw before and some cross-references. So you can see that this one, the first of them is another UniProt KB actually reviewed because it's in SwissProt. And this is the same protein name, but of that K12 reference proteome instead of the one that I was originally looking for, which was a different strain. So with that information, then I could be able to extract the information for that UniProt KB ID and then infer what that sequence is doing or the different features of that protein. So I think this is the end of the examples here. Don't worry, also you have, there's more examples lower. So we will be sharing the link to that Colab notebook there, but you have some extra snippets in there, which is one about the ID mapping flow, one about the blast flow, another one about retrieving information about PTMs, and also another one about visualizing data with a visualization library. This one is not really a biological example, so don't worry, it doesn't mean anything. It's just to show you some example of how to connect data from the API into some graphs. Let's go back to the slides. So as I said, the live demo is finished now. This is using Colab notebook, and this is the link to it. So Emily will paste the link to the chat now, and you will also have access to it later when the slides are sent to you, or yes, with the slides later on.
So we're coming to the end. I'd like to thank our funders at the NIH and the various National Institutes of Health kind of silos that help fund us there. We're also funded by EMBL, Open Targets, and SERI. We are the UniProt consortium, so we're actually UniProt is worked on by 3 different consortium sites all around the world. Me and Aurelian are based in EMBL EBI in Cambridge, Kingston, in the UK. We have a team at PIR in America and a team at SIB in Geneva, Switzerland as well. And we're broadly separated across key staff, content, and development across all 3 sites. We have loads of help and training. So we have a whole section on our homepage that's dedicated to webinars and online training. We have an ongoing webinar series throughout the year. We have tutorials and video links for our YouTube where all our recorded webinars are hosted. We also have an online course and tutorial that's in a written format. So if you prefer to learn that way, you can work your way through the written format of how to navigate UniProt. And we also have a help center and a contact us button. So if you ever get stuck or you find a protein entry that desperately needs an update, please email us. We have a help desk that's really active and we'll get straight back to you. We have a couple of upcoming webinars. So next week we've got automatic annotation systems in UniProt with Pedro. And then 16th of April, we've got curating proteins involved in AMR. That's Paul doing that in April. We've got some new features that have come to the website recently. So we've got some large-scale PTM data, that's proteomics-based data for T. gondii, the phospho modification sites on T. gondii. We've integrated the GoCam visualization into the function section of our protein entry. And then down at the bottom of the protein entry in similar proteins, we've got ortholog and paralog information for model organisms from AGR. Coming soon, we've got a new data release coming in May, so that will be 2026-02. And as part of that, we've got a lot of changes coming to our proteomes and reference proteomes. Please see our forthcoming changes, and I'm also going to put a blog in the chat if anyone wants any further information on those changes. We have a lot of programmatic help documentation as well. So if you scroll even further down our homepage, we have a UniProt data section. And if you click programmatic access on there, it will detail all our different APIs we have. We've got more than what we've touched on today because there's only so much we can talk about in an hour. Please feel free to go through all that documentation and figure out what works best for you. They're all supported by a Swagger UI in various formats too. So feel free to go and explore that. And if you do get irreparably stuck, please email us. We will get back in contact with you with a solution.
Let's get started. I think I'll take the first one, which is, I'll take the first one. So this is, if we access it from our PC or MacBook by Visual Studio Code, is memory will affect or how to handle it? I'm not too sure exactly. That might be, so if you're talking about the code here. So basically you can take that and run that anywhere. And also you will be able to download a notebook format. So if you download that locally and then run that in a local notebook, you will be able to run that on your PC or MacBook. There's no difference there. And I know Visual Studio Code can also, with some plugin, run notebooks. So you will be able to do that. But also, if you are just taking the code itself, so in Python, you will be also able to copy that code and run that in a Python environment. There's nothing specific to Colab in this code, so you should be able to copy the different cells in there and arrange that into your own script, and that should be able to run anywhere. PC, MacBook, or Linux, that would be working the same way. Okay. There's the second one, which is how can researchers ensure reproducibility when UniProt annotations are frequently updated? Are there versioned releases that should be used for computational studies? So I want to highlight one of the things I showed at the beginning, which is, so basically the API will return always the current version. So you will not be able to access previous versions through the API, that's for sure. And so to access previous versions, you will be able to use the FTP. So this is one of the ways to access data. You have some previous version in some specific format that will be available in FTP, but you don't have that customization or flexibility of searching, just like you have in the API. So an important thing you have to take into account when you use the API is really the UniProt release and the release dates here. So you can be sure that the same search will return the same results within the same release. But as soon as a new release is announced, so that will be later in the spring, you will have a 2026-02 release. So you should run your scripts again and keep your previous payload and your current payload just to be able to compare them, or if you need to reproduce them in a pipeline. So that would be the way to ensure that. I don't know, Emily, if you want to add anything on that, any advice?
Yeah, so I think if you're publishing something, include in your methodology that UniProt was searched against a specific, release. Include the release number, 2026-01. That's probably more useful sometimes than the dates because our releases and our pipeline, it moves throughout the year. So I think it's always useful to know kind of which release of which year it is. And that includes if you're downloading kind of like a set proteome, let's say you download a cluster of proteins, or you've downloaded a proteome from our reference proteomes, you're searching your mass spec data against this. Make sure you've got your release number there because obviously that can change between releases. It might change quite dramatically between this release and next release, as an example. So it's really useful. Make sure you put that in your methodology of any published papers, so someone doesn't come along and go, "Yeah, well, 3 years later I searched it against UniProt again and it's all wrong." It wasn't wrong then. It was definitely correct then against that version. And then I guess then the next question kind of feeds into this. Does the UniProt API provide mechanisms to track historical annotation changes for specific protein entries? So on the website, we've got the history tab. So you've got every release, we have a new version, a new file produced of that particular entry. So you can see we're on 01 here. That top one is going to be the current version. It was released end of January this year. And then as you scroll down, if you scroll down like quite a bit, we've got some older. Okay, cool. That's it. You see, we've got it when it was in Tremble. And then as you scroll back up, it was reviewed. You can see it was reviewed in October 2004. And if you were to click text or faster on there, you will see click text. That's probably easier to read. You'll be able to compare that to what we've currently got in the database as an example, and you'll see the changes. So you'll see that someone added several papers, and therefore they added several annotations as well. Can you do that programmatically, Aurelian?
Yeah, I just want to also highlight on the website, you can view the difference. So you select two versions, just select the last two versions. And if you do compare, you will be able to view what has changed between the two without having to do that yourself on the machine. So you have some extra annotations or new structure in that case. So this is visible there. In programmatically, what you can view is the entry version, so the current version, so here 236, or the sequence version, because the sequence might have changed also. You are able to view that in the payload. This is somewhere, I don't remember exactly where in the data that is, but you have some sequence version information, as well as the associated release number. And you are able to view that in the payload for each entry. This is you can do that programmatically. Accessing previous versions, you cannot. I don't think it is well documented at the moment. So the recommended way would be to do that manually through the website.
I think I've learned something there. I didn't know I could do that. No, I do.
Oh, even us, we learn every day.
This is it.
Okay.
So this is one for you. When retrieving protein sequences using UniProt's API for large data sets, what is the best approach to manage the data efficiency in Python without running into memory limitations?
Okay. So for that, when I said, for example, the two examples here of paginated result or stream, if you have a huge list of proteins that you're trying to download, I would think that the best, I mean, it's all up to you. You need to test and see if that works for you. But the best way to not run into memory issues would be to do that through pagination, because then you can get one page of result, which is only 25 results that you can process before requesting the next release. If you were to download, sorry, the next page of results. If you were to download the whole thing at once, you would need to obviously load all of this into memory and then process all of this, which is potentially longer also because you need to wait to have the whole result set. This is unlikely to be easy to process in a stream. That there might be ways to do that, but it's not really necessarily easy. So if you do that in page by page, it means that in your loop of processing that, you can do whatever you need in there. So if you need to compute something or if you need to write that to a file, you should be doing that in a loop as you get the result. And that way, in memory, you never get more than 25 entries at the same time. I think that would be my recommended way to do that. That's in Python, and that's the same thing for the different languages. Usually, if you do that in a loop, paginating, paginating through the results and processing as you get the result, then it would be the most efficient way to process that in a memory without affecting too much the memory. Then I think there's one for you, Emily. Well, let's see if it is. So how
Don't
does
know if it
UniProt?
is.
Yeah. Okay. So we'll have to ask the question and think about it
Yeah.
then. How does UniProt handle cases where identical protein sequences correspond to different biological functions across organisms when linking UniParc entries to UniProt KB annotations? Okay.
I've just been looking at this. So if you've got a deleted UniProt KB entry, so this is all new development for those that don't know. This is the kind of like new front end work that we've done on the website. So that this is new for us as well. If you got a deleted UniProt KB entry, you're going to get that sequence annotation page that I showed you earlier, where you've got a deleted X session and yeah, this page, right? So your annotations there are going to be specific to that particular entry and that particular species. When you're actually viewing a UniParc entry and you've got the identifiers down the left, so the UniParc entry doesn't have its annotation itself, right, Aurelian?
Mhm.
It's the other identifiers. So the identifiers are specific to the organism. So any annotations within the sequence that's called a sequence annotation page. Any annotations within there are going to be specific to that taxonomy, specific to that organism.
Yeah. Most of the time, I mean, if you're looking at the strain of the same species, um, you should, I mean, this will be automatic annotation generated, as Emily said, based on that specific taxonomy. And some of those rules are defined at a genus level or some kind of
Mm.
higher level of taxonomy so that we can ensure that the same annotations work for different strain of the same species or even different species within the same part of the taxonomical tree. And those are the rules that are defined there. So if you click there, you can see the different conditions of the rules. And that could be a way to assess if the annotation is good for you or not. And if you have a look at I wanted to show the other example when we found basically another entry. Yes, this one. So you have a annotation here. So that is the same sequence because this is the same UPI and all the cross references within the same UniParc entry will relate to the same sequence, even though they're not necessarily from the same species or the same strain. So if you have something about about a strain, and you see or you notice that you have information in UniProt about another strain within that same species, basically you will need to go have a look there and see if it applies to your strain also or not. And if it doesn't, then it could potentially be curated if you have a paper that mention the specific different function for a strain, then get in touch with us and we would be looking into integrating that into UniProt KB with that specific annotation. I think that covered everything, right, Emily?
Yeah, yeah, I think we've got there. Yeah, so a lot of this is new development that we've been working on literally the last few weeks, right? So
Mm.
this is new for us as well. I'm going to ask you this one, why I explore the other question we've got waiting for us. So how should we cite this code implementation in case we publish our studies after we've used it?
So this specific code implementation, there's no specific, you should mention that you've taken that from that specific webinar. So I think there would be a DOI and associated with that webinar. Correct me if I'm wrong, Flaminia.
Yes, you can
Yes.
cite the materials. But
So, oh
yes,
yeah, sorry, go
sorry,
ahead.
just if I can, I guess the code that you are sharing with the collab,
Yeah.
people can just use it, adapt and modify as they need. Everything will be provided under Creative Commons license. So yes.
Yes. Yes. So yeah, feel free to reuse that code and modify it. There's nothing too specific about it, so don't worry too much about that. But if you want to mention that you learned from that code from that webinar, then there will be a way to do that. Okay, so I see another one, which is can the UniProt API be integrated with tools like Cytoscape or other network analysis software for protein interaction studies? I don't see why not. I'm not too familiar with how to integrate that into Cytoscape, but this is basically you can access that data through an API, and I believe you should be able to load that into Cytoscape without too much problem. I mean, if you do network analysis specific, I mean, I cannot really tell you more about that. I don't see why you shouldn't be able to do that. But if you need specific help, do get in touch with us and then we'll send that request. I mean, as you've seen in the slide with the people that work at UniProt, there will be someone that is more focused on network analysis. So maybe we can transfer that question over to them if you have a more specific question or
So
if you're
I've
running into
I've actually
troubles.
used Cytoscape in the past.
Yeah.
Very many years ago, and I'm not gonna say I was very good at using it. So, but you can, so basically you can import UniProt identifiers into Cytoscape. Um, I'm not sure whether we're the best ones via our APIs to import interaction data. You're probably going to end up using UniProt identifiers to import from string or intact, or Mint or another interaction database. But I think essentially what you're probably looking for here is probably UniProt identifier mapping. And yeah, you'll be able to download that and download our latest release and bring that in as well. I've not in any way, shape, or form used the most recent version of Cytoscape, so probably take that answer with a pinch of salt. But yeah, if anyone's really, really stuck, please get in touch and someone will come up with an answer. Probably not me.
Okay, and we have a last one that we'll see if we will just answer that last one
Hmm.
or see if we can answer it. I see large scale proteome analysis, so I will ask you, Emily, if you know a bit more about that. So since UniPark focuses on sequence level tracking, while UniProt KB provides curated biological annotation, what are the best practices for integrating both resources in automated Python pipelines for large scale proteome analysis?
Well, there's a lot of things in that. Um, sequence level tracking or UniProt provides curated. Yeah, so I would yeah, I mean you can probably download using your UniPark identifiers. You can map them to UniProt KB identifiers and download your sequence annotation features. So this is where you've got your manual annotations mapped to the amino acid sequence. It's like what you see in the feature viewer on the website. And I would download them and map them using a line to your UniPark level annotations or mappings that you're using via UniPark. Um, for large scale proteome analysis, everything's going to be cross transferable because UniPark doesn't see taxonomy in the same way that UniProt KB does. So, especially if you're looking into specific strains for viruses and bacteria, it's going to be particularly useful to essentially map the data between the two databases, I think, going forward. For anyone that's not kind of overly familiar with UniProt in general and some of the resource I'm talking about, like the feature viewer and our sequence annotation, in the chat, I've put our webinar links, so we've got a whole series of webinars. They're all recorded. You can watch them after the fact. We've got a couple of up and coming ones as well. But there's also one in there that's a guide to UniProt for students. And even if you're not a student, it's useful because it just shows you kind of like a basic overview of everything we do on the website. And there's also a link to the webinar that was designed to be prior to this one, which is beginner programmatic access for Python. So if you want something that's looking at more basic UniProt KB queries, then please see that one as well. Yeah, I hope that's helpful.